Welcome back to the deep dive. Today, our mission is, well, it's to cut straight through the complexity of one really revolutionary piece of scientific research. We're going deep into the knowledge contained in just one paper so that you walk away fully informed on a topic that looks set to rewrite maybe some conservation history.
It really does. And this particular paper, it changes the way we look at history itself, in a way.
Yeah.
We're diving into Recovering Plant-Associated Arthropod Communities by eDNA Metabarcoding Historical Herbarium Specimens, led by Manuel Stothut and colleagues, published just this year, 2024.
Okay, so the source material here sounds, well, maybe surprisingly mundane. Old, pressed, dry plants, stuff that's been sitting in storage rooms for decades. Why on earth are ecologists and conservationists getting so excited about dusty sheets of paper?
Yeah, it sounds dry, literally. But herbaria, these natural history collections, they're actual treasure troves. They contain millions of historical plant records from, well, nearly every corner of the globe. For maybe 150 years, they've been used to study the plants themselves. Fair enough. But the real significance, the kind of gold standard knowledge we've been missing, is the historical record of plant-arthropod interactions.
Right. And that matters because, well, the big story in ecology right now is this widely reported global arthropod decline. We're talking insects, spiders, mites, the tiny engines of pretty much every terrestrial food web.
Exactly. And if we want to understand if these declines are real, how steep they are, maybe why they're happening, we desperately need time series data. We need to know what was happening 20, 40, maybe even 60 years ago. But, standardized long-term monitoring data for insects, it basically just doesn't exist for most places.
So the core question these researchers are tackling is actually pretty profound, isn't it? Can these pressed plant specimens collected long before anyone was even thinking about insect DNA serve as kind of time machine? Can they reconstruct entire insect communities from the past?
It almost sounds like science fiction. But the method is really elegant. Environmental DNA or eDNA metabarcoding. In simple terms, when an arthropod interacts with a plant, walks on it, chews a leaf, leaves behind microscopic stuff like feces, saliva, maybe shed skin, it deposits DNA traces. The study hypothesized that the simple process of drying and pressing the plant, which is meant to preserve the plant itself, actually preserves these tiny DNA traces too.
Okay, so let's see how robust this technique actually is. They didn't just grab one sample, right? They set up experiment 1, testing historical herbaria across different ages and geographies. How diverse were their source materials?
They chose 3 very distinct collections. The youngest was a 6-year-old institutional herbarium from Iran, featuring a desert plant called Calotropis posera. Then they sampled 2 private collections from Germany, one 18 years old, and a third that was remarkably 60 years old, collected back in 1963. That age range was really critical to see how long this DNA signature could actually survive.
And the headline finding from that first test
Yes.
is a success, a big one.
Yeah, a resounding success. They recovered arthropod eDNA from specimens up to 60 years old.
60 years, wow.
Yeah, and they recovered 245 unique ZOTUs. That's probably a term we should unpack for a moment.
Please do. Yeah, for listeners, what exactly is a ZOTU, and why does finding 245 of them matter so much?
Okay, so think of a ZOTU that's a zero-radius operational taxonomic unit as an extremely granular genetic fingerprint. They're essentially distinct haplotypes, sort of like unique genetic variants. So we were looking at humans. Finding 245 ZOTUs would be like finding 245 genetically distinct populations or closely related species of arthropods. This isn't just a generalized insect presence signal. It's like a high-resolution map of the historical community.
That level of detail is phenomenal, really, and the diversity of what they found, that tells an interesting story too, right? About who was actually using the plant.
It absolutely does. They recovered DNA from 10 different orders of arthropods. It spanned every major ecological role you can think of. Herbivores like gallers and leaf miners, sure, but also predators, parasitoids, even pollinators. It really is like a complete snapshot of the plant's interactome at that moment in time.
I noticed the breakdown showed that sap-sucking arthropods were the largest group, 39% of the taxa they found. Does that specific ratio tell us something about the historical ecosystem, or is that maybe just an artifact of how those particular insects leave DNA behind?
That's a great question. It's likely a bit of both, actually. Sapsuckers, like aphids or cicadas, they spend a lot of time physically on the plant. They leave behind pretty robust traces like saliva injected into the plant or honeydew. In contrast, pollinators, which were less than 1%, they just don't stick around long enough, especially once the plant is collected and pressed, to leave much DNA. So yeah, the ratio is probably skewed towards organisms that have really intimate long-term contact with the plant tissue.
Okay, here's where the conversation gets really interesting, I think. Because any skeptical scientist, myself included maybe, would immediately raise a red flag and say, hang on, isn't that just contamination?
Yeah.
Storage pests get into old archives all the time. Silverfish, book lice.
Absolutely.
Yeah.
And that is the most critical barrier the authors had to overcome. They knew this, so they instituted really rigorous validation checks. The first one was contamination control, specifically identifying and removing what we call synanthropic pests.
Which
Basically pests that thrive alongside humans, things like common storage beetles, silverfish, certain moths that you often find in archives or even homes. They identify the DNA signatures of these known pests and computationally strip them out of the data set. Interestingly, the professionally managed institutional herbarium had less pest DNA, only about 5% of the DNA reads, compared to the older private herbaria, which had up to 15%.
Okay.
Which kind of suggests that professional curation and maybe treatments help, but yes, some accumulation of background pest DNA is probably reality over decades.
But the contamination question gets really nailed down when we look at the geographic origin of the non-pest DNA, doesn't it?
It really does. This was their validation check number 2, geographic origin. The recovered DNA of the actual plant-associated communities perfectly reflected the place the specimen was collected. For instance, the Iranian samples. They contain oriental species, things like the parasitoid wasp Saccharicola, these you expect there. Meanwhile, the German samples contain typical European taxa, like the gallmidge Asphendilia cerathamni. If this were just random lab contamination or even general archive dust, you'd expect to find these species mixed up, or maybe common lab contaminants, not these geographically specific creatures.
So that's pretty convincing. But they took the investigation even one step further, didn't they? To establish specificity, they wanted to prove the DNA wasn't just, like, floating around the storage room from one specimen to another.
Exactly. That's where validation check 3 comes in. Specificity. They compared different plant species stored right next to each other, literally in the same herbarium cabinet. And they found hardly any shared arthropod taxa between them. They quantified this using something called the Jacquard Dissimilarity Index.
Okay, stats time. For us non-statisticians, a Jacquard dissimilarity of approximately 0.99, that sounds incredibly high. What does that number tell us?
It means there was virtually zero overlap, like 99% different. The genetic signature detected on plant species A was almost entirely different from the signature on plant species B, even though they were stored side by side. If the DNA had just been floating around the collection room and settling randomly, you would have seen a lot more shared taxa. The fact that they didn't, it proves the DNA signature is tightly bound and really specific to that individual plant specimen.
They even showed that specificity on a micro level, didn't they? Finding different insect communities on different parts of the same plant like flowers versus leaves on those Iranian specimens?
Yes, and this leads to what I think is the ultimate aha moment, detecting these incredibly specialized ecological interactions. They didn't just find random bits of DNA, they recovered evidence of highly specialized relationships. For example, the herbivore Lamiogethes difficilis was found exclusively on its known host plant, Lamium album. Only there.
That's incredible complexity preserved just sitting there on a dried leaf.
It gets even better. They even recovered a tritrophic interaction. That's 3 levels of the food chain, all preserved on one dry piece of paper. They found the aphid Cavriella egopodii and its specialized parasitoid, a tiny braconid wasp called Benodoxys brevicornis, both found together on ground elder.
Wow.
This proves that herbaries aren't just archives of plants, they're effectively archives of ecological dynamics, whole interactions frozen in time.
That level of detail really proves these collections are true time capsules. But the authors knew the next big hurdle was translating this potential into the urgently needed long-term ecological data, which brought them to Experiment 2, the time series analysis.
Exactly. Moving beyond those isolated historical snapshots, they tested the method's utility for actually monitoring long-term change. For this, they analyzed archived leaf samples, European beech leaves, from a standardized German forest monitoring project. And this spanned 2 decades, from 2004 right up to 2021.
Okay, so what did the 20 years of time series data tell them about arthropod health in that particular German forest? Was it decline?
Well, the picture was nuanced. The data showed quite pronounced fluctuations in arthropod richness over the 20 years. Notably, there's a considerable diversity increase between 2004 and 2006, which was then followed by a drop, and after that, relative stability right through to 2021.
Huh. That stability finding, particularly in a forest environment, seems to contrast sharply with some of the headline-grabbing declines we often hear about, especially from heavily used farmland or grassland areas. What does this method add to that ongoing, sometimes quite heated debate about insect decline?
I think it suggests the complexity of the issue, that decline isn't necessarily uniform across all habitats. We often hear these intense reports of insectageddon, right? But the stability they found in this forest study, specifically deep in the canopy, mind you, suggests that forest systems might have different dynamics, maybe longer fluctuation cycles that just aren't captured by shorter-term surveys in other habitats. This herbarium eDNA method allows us to potentially put those intense, localized declines into a much broader, longer temporal context. It adds nuance.
And by looking at individual species trajectories within that forest, they could see differences even within the same study area, right? Not just overall richness.
Absolutely. For example, the common beach gall midge, Miciola fagi, seemed pretty stable over the study period, but another species, the leaf-mining beach pygmy, Stigmella hemargirella, appeared to actually be declining in their samples. This kind of granular species-level data over time is exactly what conservationists and climate scientists need to create really targeted action plans. You can see who's struggling.
They also performed a third experiment. Experiment 3, storage stability. This seems critical, right? To make sure that the process of being stored for decades didn't just immediately destroy the DNA or completely change the picture.
Yeah, super important validation step. They compared fresh samples to samples that were treated in different ways, freeze-dried and cold-stored, or silica gel-dried and kept at room temperature. And the crucial finding was that standard dry storage at room temperature, which is precisely the condition in most herbaria, for 6 months, did not significantly alter the recovered arthropod community richness or composition. It turns out those dry, dark room temperature conditions are actually, well, pretty excellent for preserving arthropod eDNA over moderate timescales.
However, they did note a limitation, a critical one, regarding DNA integrity and age, particularly with those really old samples.
They did. While the eDNA was generally stable, those 60-year-old specimens showed somewhat reduced diversity when they tested them using a method designed to amplify a longer fragment of DNA. This suggests that the DNA does begin to degrade, to fragment into smaller pieces over very long timescales. But importantly, that degradation seems minimal in the first few decades. What this tells us, practically, is that future researchers should probably focus on amplifying shorter DNA fragments to maximize what they can recover from the very oldest historical records. Adapt the method to the age.
Right, makes sense. Before we wrap up the implications, let's quickly touch on the necessary sampling method, because this is important for curators who, guard these valuable specimens. They needed a slightly destructive approach to get the best data, correct?
They did, unfortunately, for pristine preservation purists, perhaps. To access the true ecological signature, the DNA left deep inside galls or in leaf mines or from saliva and puncture marks, they had to actually homogenize, basically grind up a few leaves or flowers. They did test non-destructive washing methods, like spraying water or using a paint roller lightly, but those methods primarily recovered the DNA of those synanthropic pests we mentioned earlier. About 73% of the DNA reads were just storage pests. Yeah. So the really valuable plant-associated community data seems to be bound up more tightly within the preserved plant tissue itself. You need to get inside.
Okay. So stepping back, what does this all mean for the bigger picture? If you're a biodiversity scientist or just someone who cares about conservation, why is this method potentially a game changer?
I think the core meaning is this. Every single one of the millions upon millions of plant specimens housed in herbaria around the world now potentially preserves an accurate snapshot of its interacting arthropod community, its interactome, at that specific point in time it was collected. This just promises unprecedented insights into how these critical plant-arthropod interactions have shifted over decades, even centuries in some cases.
It seems like it could effectively revolutionize our ability to monitor changes over space and time, tracking the emergence of invasive pests, or maybe confirming the decline of highly specialized species over scales that were just impossible before. It feels like it closes a really critical knowledge gap in understanding how biodiversity is assembled and maybe disassembled in the Anthropocene.
It really does. But the authors wisely note a final important caveat. The full power of this analysis lies not just in looking at these historical snapshots in isolation. It's really about comparing them robustly to present-day food webs, reconstructed from fresh plant material using similar methods. That direct past versus present comparison is the key that unlocks the true measure of decline or stability or change.
Okay, so to bring it all back home for everyone listening, the core takeaway is that these historical plant collections, maybe long overlooked for their insect interactions, are actually incredibly detailed, deep storage time capsules of entire ecological communities now accessible via this eDNA metabarcoding technique.
Exactly. And that leads us to, I think, a truly provocative thought for you to consider as we finish up. If a simple pressed leaf collected by some dedicated botanist, maybe back in 1963, can hold such a detailed 60-year record of an entire ecosystem's hidden life, the specialized predators, the prey, the parasites all interacting, what other seemingly innocent or mundane historical archives, perhaps things we aren't even systematically cataloging today, might be out there, just waiting to reveal critical data about biodiversity change? The past, it turns out, is now directly informing the future of conservation in ways we never quite expected.